Okay, so here we have a cell, and we've got it's surrounded by a cell membrane, goes all the way around the cell, and here's the nucleus, which houses the master copy of the instructions for making things in a cell. Here's the nucleolus that makes those little guys called ribosomes. Here is the smooth ER, which is a hallway attached to the nucleus. Here's the rough ER, which is also a hallway attached to the nucleus, but we see there are ribosomes here. And because ribosomes make stuff, that means that the rough ER is going to be making stuff. And here are vesicles. They're containers. They're carrying things throughout the cell. Here are the Golgi bodies or Golgi apparatus. And what they do is they check to make sure that whatever the vesicles bring them is made correctly. And then they address, it's kind of like addressing an envelope. And then if it's correctly made, correctly addressed, the vesicle then goes off and can either go out of the cell or stay in the cell. If whatever the Golgi bodies is checking is not correctly made, then the vesicle heads over to a lysosome, and the lysosome breaks it down and recycles all the pieces that can be used to remake something again. And lastly, here's a mitochondria, which converts sugar to energy that the cell can use to do all this. So now the question is, how does the cell membrane control what is allowed in the cell and what is allowed out of the cell? Okay, so let's take a look at some examples here. Here's the door to a building. When a door is open, a human can easily walk in and out of the door. So can a cat, so can a dog, so can an ant. A fly can fly in. It's a pretty big opening, right? Here's a window, little smaller. Yeah, a human might be able to fit through the window. Certainly a dog or a cat or a bird or an insect can go through the window, such as air, but it is smaller, okay? So that means that really big things, like an elephant, would not be able to fit through that window. If we put a screen on the window, now, we're not talking about pushing the screen open like a burglar or anything, but screens will keep humans out, dogs, cats, birds, bugs, but air can still go in and out through the screen. So it all has to do with the size of the thing that wants to come into the cell or out of the cell. The size of the item has a lot to do with it. And here we see a close-up of a window screen. It's got to be a really small substance to go through this window screen. Well, these guys here, called phospholipids, they are the molecule that makes up the cell membrane. So we're going to learn a little bit about them. It's called a phospholipid because the round part is called a phosphate head, because there's an atom of phosphate there, along with some other stuff, but basically it's basically called phosphate, a head. And there's a tail on it, or two tails actually. We just simply call it one tail for simplicity's sake. And it's called the lipid tail. Lipids are fats. Okay, so now think about fats and oils and, okay? Well, it turns out that the phosphate heads of the phospholipid, they love water. They love being really close to water. So this round portion here loves being next to water. But the lipid tails, now lipids are like fats. The lipid tails absolutely hate water, can't stand being near water. Okay? Well, these guys are attached to each other. So that this can get kind of interesting if we've got a couple phospholipids hanging out with each other. It gets really interesting because we know that oil and water don't mix. So how is this guy going to behave in water or with other phospholipids? We're adding a little bit of vocabulary here. So the phosphate head, we know, loves water. The lipid tail hates water. Another word that we use for loving water is hydrophilic. Hydro means water, philic means to love. think of Philadelphia, the city of brotherly love. The lipid tails down here hate water. They are hydrophobic. Hydro means water. Phobic means to be scared of. You don't like it. You're scared of it. So I have acrophobia. Acro is height. I'm scared of heights. Okay? Another term that we can use instead of hydrophilic is polar. Another term that we can use for hydrophobic is nonpolar. You'll see both those terms. Okay? So basically, if something loves water, it's hydrophilic and polar. If it hates water, it's hydrophobic and nonpolar. So let's say we've got 4 phospholipids hanging out. How are they going to arrange themselves? Each one of them wants to sit next to their friends. So the phosphate heads want to hang out together, and the fatty acid tails, the lipid tails, want to hang out with each other, but the heads and the tails don't want to hang out with each other. Hopefully that makes sense. And this is how they do it. We can see here that this phospholipid is next to this one, which is next to this one, which is next to this one. The heads are all close to each other because all 4 of these love water. The fatty acid lipid tails here are all close to each other because they hate water. So these 4 little guys are hanging out with each other. These 4 guys are hanging out with each other. This will never happen because look, here we have lipid tails that hate water next to a phosphate head that loves water. This is not going to happen. This is what we see instead. So, this is what we end up with. These phosphate heads are all in line. These lipid tails are all in line. And check it out. These tails flip around. So that means that these tails all hate water and are all hanging out together. There are no heads in the middle here because they love water. Uh-oh. Uh-oh. So what's going to happen? These guys are already all set up. They're sitting there at the cafeteria table. And here comes someone who wants to join them. We have to figure out how they're going to join them without disrupting the arrangement here. Because we know that this phosphate head that loves water can't hang out here next to these fatty acid lipid tails because they hate water. So we're going to have to figure something out. So here's the solution. What they do is they actually bend around in a circle. That looks like a cell. So here we have the inside of the cell. Here we have the outside of the cell. And the way these guys are lined up, the tails are all together and next to each other. So there's no water in between these tails. And these heads are all sticking out away. These heads are sticking in a way and they're all next to each other. So everyone is happy. So here's the inside of the cell. Here's the outside of the cell. And this forms a double layer. We actually call it a phospholipid bilayer, like bicycle or binoculars. Bi means 2. And it works out really, really well because on the inside of the cell is cytosol. Remember, that's like a watery jelly-like substance that takes up space where all the chemical reactions or most of the chemical reactions occur within a cell. So there's water inside the cell, which is awesome because these phosphate heads love water. They're hydrophilic and polar. And it's also great because outside the cell, there's water. And again, we have the phosphate heads are out here facing the water. They are hydrophilic, polar, and love water. So they're happy. And look at these guys. These lipid tails that hate water, they're hydrophobic and nonpolar. They're all hanging out with each other. There's no water in between them at all. And that's how phospholipids assemble on their own in this kind of a pattern to form a cell.